Right, thank you so much for the opportunity to pitch. I'm Bhavanjeet Brar. I'm the CEO and founder of JAAN Biotherapeutics. We are regenerating heart muscle to treat cardiovascular diseases by targeting microRNAs. So in this room, 44% of the women here have some form of cardiovascular disease. And in 2019, cardiovascular disease was the leading cause of death of women. This is with all cancers combined and lung disease. Females represent 48% of deaths from cardiovascular disease, and 9.1 million females have chronic heart disease in the United States. 3 million of those patients got that disease after having a heart attack. Heart disease is the number one cause of death globally of women. It can be caused by a heart attack where there's a blockage in a blood vessel that delivers blood and oxygen to the heart muscle. This results in permanent heart muscle death. The heart becomes weaker, heart failure develops, and the patient dies. We know that in the US, 20% of females that will have their first heart attack will die within the first year of that heart attack. There are certain risk factors associated with that we'll go into. The current therapies do not regenerate heart muscle. Attempts to regenerate heart muscle have failed. Our technology can achieve this. Exogenous stem cells have not been commercialized. There have been over 90 clinical trials, billions of dollars spent, and nothing moved forward into commercial. There are approved drugs. They are unable to regenerate heart muscle. They just manage the symptoms. We know that there are tens of thousands of women on the heart transplant list here in the United States, but only 3,000 heart transplants every year. There are hundreds of pharmacological agents, peptides, gene therapies, and I've spoken before about one company developing microRNA drug, but that has shown limited efficacy in clinical trials because none of these drugs regenerate heart muscle, which is the holy grail of cardiovascular drug development. Our solution is technology that we've exclusively licensed from the Salk Institute. So in this study that they published in Cell Stem Cell in 2015, they showed that if you inhibit 4 microRNAs or non-coding RNAs using 2 viral vectors delivered into the heart muscle of mice with permanent heart attack, you can restore heart function 90 days post-heart attack. That's measured by ejection fraction. The baseline is about, in heart failure in this model was about 40%, and it reverted back to normal, which was about 60%. And it reduced scarring in the left ventricle by 60%. So lowering the levels of the microRNAs can regenerate heart muscle by activating proliferation and cardiomyocyte dedifferentiation pathways. So we have 2 strategies. The first is a viral therapeutic approach that we've developed. This is called JBTMR2. It's cardiotropic, like the heart, doesn't integrate. That's the beauty of expressing RNAs. They're transiently expressed. They don't change the DNA, so there's less potential of lethal side effects. It's formulated for IV administration, and it's given at a low dose intracoronary or intravenously. We've also developed synthetic oligonucleotides that are stable in the blood as an alternative to viral therapeutics, and they can be given subcutaneously. So this is our development pipeline. So we're solely focusing on microRNAs. Our lead therapeutic is JBTMR2. Our patents are allowed. So we have 5 US patents allowed, which will allow us to also regenerate heart muscle for other diseases where cardiac muscle regeneration is required, such as cardiomyopathy associated with Duchenne muscular dystrophy. So this is the pig model that we set up at UCSD. So this is the model that we set up at UCSD. It's a line model of cardiac injury. And this is what happens in the pig model following we make a heart attack. So this is the left ventricle, which is a main pumping chamber of the heart that pumps blood and oxygen to the heart muscle. You can see at baseline it's pretty small, but after the heart attack, it doubles in size. The area of injury is shown here in dark blue. This is similar to the pathophysiology that actually occurs in human heart failure. We conducted a proof of concept study in pigs with a heart attack. We dosed the virus twice after the heart attack and then 2 months later, and then followed the pigs for 3 months. And we were able to normalize heart function, which is measured by ejection fraction, and we used cardiac MRI to do that. So we increased left ventricular mass as measured with cardiac MRI. So with any cardiac regeneration therapy, you should show an increased left ventricular mass because you're building muscle, and the drug was safe. The evidence of safety has been extensively evaluated across 7 preclinical studies. Transiently express, they're cardiotrophic. We have no abnormal measures. Regeneration is specific to the heart muscle, and we've yet to conduct GLP toxicology study. This is just an assay where we looked to see where the virus is going in a mouse study. So we're seeking $25 million to complete our clinical trials. The first tranche is $5 million. That's to conduct our GLP toxicology studies and file our IND. The second tranche of $10 million to manufacture our virus, and $10 million to execute the clinical trial. We're first targeting patients that are high risk for poor outcomes, and so these are the risk factors. So 400,000 women have heart attacks every year in the United States. Those that have risk factors such as they're over 65, diabetes, low ejection fraction, and as spoken before in the previous talks, present late to ER following a massive heart attack. They are likely to not make it, 50% dying in the first 30 days following the heart attack. That is a multi-billion-dollar market for women alone. And it also affects men. So the first product will be administered in the catheterization lab or shortly after the heart attack in a hospital-based setting to prevent rehospitalization. The second product will be an outpatient product, and that's with patients with established ischemic heart disease measured normal imaging and administered by the cardiologist. That is a multi-trillion-dollar market. The leadership team has over 200 years of experience. I'm the CEO and founder of the company. I've developed 2 drugs for heart failure from the bench to clinic, onto completion of phase 2 clinical trials for heart failure, where I manage that clinical trial. Dr. Bader is our microRNA consultant, and Professor Kirk Peterson is the interventional cardiologist who conducted all of our animal studies double-blind at UCSD and all the data analysis. We have a network of key advisors as well. We have long-term patent protection. We have 5 US patents issued that cover our targets, composition of matter, over 30 drugs that we have, and how to give our drugs, and with additional filings in process. This is just some of the liquidity events in the cardiac space, and it's just increasing, significant unmet medical need, which is going to increase. These are involving cell therapy, gene therapy, and also RNA technology. To summarize, regeneration of heart muscle is the holy grail of cardiovascular drug development. Ischemic damage is the main cause of death of women in the United States. Attempts to regenerate heart muscle failed. Our solution is by targeting 4 specific microRNAs using oligonucleotide approach or also a viral approach with excellent safety to date. We're seeking a $25 million Series A in tranches to get definitive efficacy and safety in pigs before we test in man, and then execute our clinical trials in patients at high risk. Thank you very much.
Thank you, Rar. Do we have a question? All right. Thank you so much.